Hi, Sister Mayo. I'm Catherine, and this is our lesson 10 group problems. So, yeah, here you go. Number 1 is asking, suppose a eukaryotic messenger mRNA tight sequence RNA polymerase 2 produce this molecule from left to right. Else, which end of the mRNA molecule is the 5' end and which one is the 3' end? So for A, 3' end, and it goes UGC, UUG, CCAAA, UCCC, GAUA, GA, and IMEND. For B is asking us what is the sequence of the template DNA strand, include the polarity. The DNA strand starts with a 5' ends with a 3'. The nucleus, the sequence of the template, it's ACGAA, CGG, TTT, AGT, ACTA, TCT. For C, what is the sequence of the coding DNA strand? 3' to 5', and the sequence is TGC, GCCAAA, TCAT, CCATAGA. Asking us to draw an image that shows the 3 nucleic acid above during the elongation stage of transcription. That's what I drew. And then we see that this is the mRNA and this is the template strand, and it's from 5 to 3, the coding strand. The mRNA is from 5 to 3 as well, and the 3 to 5, and at the beginning, you see the transcription plate in the top box. I wrote it on the side here. For number 2, as to fill in the blanks. A is TFIID, the top box, while the sigma factor binds directly to the -10 and -35 sequences. For B is guanylyl transferase, I don't know if I'm saying that right, is a eukaryotic enzyme that links the 5 carbon of one nucleotide to the 5 carbon of another nucleotide. For C, group 1 introns and group 2 introns is the nucleoside guanosine as a cofactor during catalysis, while group 2 introns use adenine as a cofactor. CPSF is an endonuclease that assists in terminating transcription in eukaryotes. And for E in lesson 9, we learned about general transcription factors. That's TFIIE is a GTF which acts as a bridge subunit to link TFID to TFIIF RNA polymerase 2. IIH is a GTF which acts to phosphorylate and activate RNA polymerase 2. For F, the spliceosome is composed of subunits called SNRNPs. Spliceosome subunits are composed of 2 components, proteins and snRNA. And you can find the answers to all of these in the instructor notes. This question concerns the proteins involved in transcription and RNA modification. So for A, indicate 2 proteins in either prokaryotes or eukaryotes that act as a helicase to separate 2 DNA strands during transcription. TFIIH and RNA polymerase. For B, indicate 1 protein in prokaryotes that acts as a helicase to separate RNA from DNA during transcription. that the row P. For C, indicate 3 molecules in eukaryotes that function during RNA modification as ribosomes. And that's S and R and P, group 1 intron and group 2 intron. For D, indicate 1 protein in prokaryotes that act as a transcription factor. And that is the sigma factor. And for E, in eukaryotes that synthesizes a long string of identical RNA nucleotides, a DNA strand for RNA synthesis, and that is the poly-A polymerase. Question 4 is asking us to draw the images of the resulting nucleic acid structures following the hybridization experiments described in parts A through D below. So part A, mRNA for YFG hybridized to the genomic YFG. So these are my little drawings. But for A, we see the introns and then the splicing that's taking place and that the introns are cut off. So this is the hybridization, mature mRNA. In the hybridization, these are the loops that would be left. You would have 4 intron loops on the mature mRNA, a loop, the mRNA, and the DNA strand right here. The mature mRNA for YFG is hybridized to the genomic DNA copy of YFG. So in this one, you have the 3' end to 5' end is the coding template strand. Is the template. And I have 1, 2, 3, 4, 5 R loops and 1, 2, 3, 4 intron loops. And you can see here that the splicing is taking place. So for part C, the mature RNA for YFG is hybridized to the genomic DNA copy of YFG. Most intron in the pre-mRNA contains a defective donor site and branch site. I assume that the introns are removed using this spliceosome method. So for part C, we have here the defective donor, and then the spliceosome. The introns are being removed by the spliceosome, and it's from 3 to 5. Sorry, yeah, it's the 3 to 5 coding the mRNA strand, and the 5 to 3 the DNA template. And then you have 1, 2, 3, 4 R loops, and then just 1, 2, 3 intron loops after everything is done. And then for part D, mRNA for YFG is hybridized to the genomic DNA copy of YFG. In this case, the second and fourth introns in the pre-mRNA redirection both contain defective donor site and branch site adenine. So assume that the introns are removed using the spliceosome method. So for D, and then we have the 3 to 5 prime, and that's the coding strands, and we have the 2 defective. Sorry, we have the second and the fourth defective donor sites. And then we have the mRNA template, the mRNA strand that's from 3 to 5, as well as the coding strand, which is 3 to 5. And then for the template, it's from 5 to 3, and you have, at the end of everything, you have R loop and 2 intron sites. For part 5, for part A, which of the above is found in the bacterium Escherichia coli? RUT, -35, and Newsom. For B, which of the above is found in the yeast Saccharomyces cerevisiae? I totally butchered that. And I have negative 25, enhancer, group 1 intron, CJ box, GC box, TF2D, SNRNP, and the silencer. For C, which of the above is involved in the transcription initiation? I have negative 25, enhancer, negative 35, GC box, TF2D, and the silencer. TFIID, sorry, and the silencer is involved in a transcription termination. And we have RUT and NUSA. Which of the above is a DNA sequence? And it's negative 25, enhancer, negative 35, GC box, and the silencer. Thank you, Sister Mayo, and here's my group work.